So this is our mitosis sort of simulation. This outer circle is representing my cell membrane, and this inner circle is representing my nuclear membrane, and this little blue pom-pom is representing a centriole, okay? So a centriole is going to help with the cell division process. It's an organelle or a part of the cell that does that. So when the cell's in interphase, where it spends sort of 90% of its time growing, performing normal functions, you can't actually see the chromosomes that are in the nucleus, but they are there. They're in a really thin form called chromatin, and they're really hard to study at that point because they're very difficult to see even with a powerful microscope. But if we were to sort of zoom in on what was there, we would see really thin versions of chromosomes. And like that, okay? And at the very end of interphase, we get what we call the S phase, and that's when they replicate. So they actually double. Again, we can't really see them under the microscope like this until prophase, but make them match. But if we really were to be able to see them, that's they would look like thin versions of this. And in the S phase, the centrioles also replicate, because again, the goal is to make two new identical cells. So the DNA has to double so that when the cell splits, you end up having two cells with the equal amount of DNA that you started with. So once interphase is done, we enter prophase. And in early prophase, you can see this under the microscope a little bit. So in interphase, the nucleus sort of looks like all one color if you were to look at a high school microscope. But once you get to prophase, you start seeing sort of dark splotches, and those are the chromosomes that are starting to thicken. Some will have more dark splotches, some will have less, because every cell is undergoing mitosis at a slightly different time. They're not all doing it at the exact same time, so no two cells will look exactly the same if you take sort of a screenshot of what mitosis looked like at that point. But as we go through prophase and they do thicken up and we can see them and study them, we do see that they sort of look like these little X pairs here. And in terms of vocabulary, sometimes you will hear these called chromosomes. Sometimes you will hear them as chromosome pairs. And sometimes you'll hear them called two chromatid chromosomes. So usually in this phase, we're calling them like two chromatid chromosome, but to keep it simple, let's just call them chromosome pairs or chromosomes. It doesn't really matter. We can study them no matter what they're called. But as prophase goes on, a few other things happen as well. One is that the nuclear membrane starts to break down. And so obviously if we need the chromosomes to move to different sides of the cell, they have to get out of the nucleus. And the centrioles also start moving towards the edges of the cells as well. Okay, so again, if in early prophase, they might be close together, and then as prophase goes on, they get further and further away. And as they get further and further away, they start forming these like spindly things around them. They're actually called spindle fibers, that are going to help with cell division. Okay, so what's eventually going to happen is they're going to move to opposite sides, and the chromosomes are sort of going to move to opposite sides too, like this. And this is when we enter metaphase. Okay. And the chromosomes are going to be lined up and attached to the spindle fibers, sort of one holding each. Okay. Chromosomes are currently attached together through something called a centromere. Sometimes we use toothpicks to show that they're like stuck together. I didn't this time just to keep it simple. So the very sort of spot where they're attached is called the centromere, and that's going to eventually break at the end of metaphase. So metaphase, think middle. The chromosomes are along the middle, also known as the equator, just like on Earth. And then the centrioles are at the two ends of the cell, which we call the poles, just like on Earth. So it's an easy way to remember. Middle is the equator, ends are the poles. I'm just trying to get them all to attach. You get the idea. So this is our metaphase. In anaphase, what's going to happen is the spindles and the centrioles are going to start sort of pulling towards the end. So that means that our chromosomes are going to start splitting at the centromere and start making their way towards the poles. So the spindles are actually going to pull them. Obviously, I can't simulate this all together, but as they move, they pull the chromosomes with them. And now they're single chromosomes. They're not paired up anymore. Okay, like this. And what's actually happening at the same time is that the cell is starting to stretch as well. So you'll see it looking a little bit sort of like an oval during this. And obviously some will go faster and some will go slower. And eventually they're going to reach the ends. And when they reach the ends, the spindle fibers start to break down because they're not really needed anymore. Chromosomes start moving together towards the center. Spindles are gone. And the nuclear membrane starts to reform around them. They want to be protected again in the nucleus. So we get a nuclear membrane forming around this side. And longer one. Nuclear membrane starting to form around this one. Okay. And this is telophase. So once the chromosomes are in the nucleus, they're comfortable, this starts reforming, they start to thin out again and become chromatin. And once the DNA is sort of safe in the nucleus, all the other organelles will split as well. And that's called cytokinesis. So the vacuoles and the endoplasmic reticulum and the ribosomes and the Golgi apparatus, all of those start to split now too. And that makes sense because we need the brain of the cell to sort of be intact. Everything else can move. And then the cell really starts to get ready to split. So it's actually going to pinch in, and that pinch is called the cleavage furrow. And eventually it's going to cleave off. That's why it's called cleavage furrow, because it's cleaving is sort of like that breaking. And it's going to create two new cells. And this obviously would break completely. Let's see if I can cut it without breaking everything. And we'd have sort of our two cells. All ready to start the whole process of interphase again. So the original cell we now call the parent cell, and these are called daughter cells. So they're genetically identical in every way, despite my terrible artwork, but they are a lot smaller. But again, interphase is a time of growth, and so they will grow as well. So those are our phases of mitosis. So again, interphase is your growth phase. It's not really a part of mitosis. It's just the regular cell life. And then our mitosis phases are PMAT, so prophase, metaphase, anaphase, telophase. Cytokinesis happens after. Again, it's not really part of mitosis, though a lot of diagrams show it happening together. So telophase first, then all the other organelles and cytokinesis, and then interphase starts all over again. And the rate of mitosis will happen differently depending on the type of cell. So this cell had 8 chromosomes or 4 chromosome pairs before it split. Humans have 23 pairs or 46 chromosomes before they split. So think about sort of what organisms might have 4 chromosome pairs versus 23 chromosome pairs when you're doing your research.